So picture this. You pull a carton of milk out from the back of the fridge, and, you give it that nervous little sniff test.
Oh, yeah, we've all been there.
Right. But in that exact same moment, maybe halfway across town, there's a doctor giving a patient this really militant, almost aggressive lecture about making sure they finish every single pill in their antibiotic prescription, even if they feel perfectly fine.
Which they usually do after a few days.
Exactly. And these seem like two completely unrelated scenarios, right?
Yes.
Separated by miles, completely different situations. But they are actually governed by the exact same invisible mathematical law.
Yeah, the underlying mechanics of both of those situations rely entirely on this microscopic timeline, one that most of you listening probably never even think about. Yet it dictates, so much of our daily survival.
And that timeline is exactly what we're focusing on in our deep dive today. We're digging into a really fascinating stack of sources, some detailed microbiology notes, a few historical research papers, and they all center on one monumental concept, the bacterial growth curve.
Right.
Our mission for this deep dive is to decode this classic biological model. We want to show you that the famous lag, log, stationary, and death phases aren't just, abstract ideas you might vaguely remember from some high school biology class.
Definitely not.
No, this curve is literally the invisible architect of our food safety, our medicine, and even huge global industries.
And, when you look at the growth curve on paper in these sources, it looks deceptively simple.
Oh, totally. Just a line on a graph.
Right, a line goes up, it flattens out for a bit, goes down. It's just a tidy little hill. But that smooth line is actually masking this chaotic, microscopic war zone.
A war zone. I love that.
Because the biological reality of this curve is full of unexpected surprises, highly evolved dormancy, and honestly, some mathematical impossibilities.
Okay, let's unpack this, because before we actually get into tracing the path of the bacteria through those phases, the sources are very clear that we have to define what this curve is actually measuring. Because there is this massive misconception right out of the gate.
Oh, huge one, yeah.
Right. So, strictly defined, the bacterial growth curve is a graphical model tracking the size of a bacterial population over time. And this happens in a closed system, which biologists call a batch culture, meaning it has finite nutrients.
Nothing goes in, nothing comes out.
Exactly. You plot time on the x-axis and the logarithm of the viable cell number on the axis. But the trap, the one almost everyone falls into, is looking at that curve and thinking it describes the life cycle of a single bacterium.
Right. People assume it represents one cell experiencing, birth, maturity, old age, and then death.
Like a human life.
Exactly.
Yeah.
We constantly anthropomorphize the microscopic world. Even professional microbiologists back in the early 20th century treated these phases as if they were physiological states that the entire culture just enters into at the exact same moment.
Like some synchronized swimming routine?
Yeah, but a single bacterial cell simply doesn't experience a lag phase or a death phase the way the macro level curve implies. A single cell only really has 3 distinct options at any given second. It divides, it doesn't divide, or it dies.
Okay, so the best way to visualize this for you guys listening is to think of a massive traffic jam. If you look at a highway from a helicopter, you can track the overall trend perfectly, right? Traffic is stopped, then it gradually accelerates, then it reaches cruising speed.
Right, the macro trend.
Exactly. But that macro trend doesn't mean every single individual car is hitting the gas pedal at the exact same millisecond.
Of course not.
One person is totally stopped, someone else is switching lanes, maybe someone else is pulling off onto the shoulder. The smooth curve we see on the laboratory graph is really just an emergent statistical signature.
Yeah.
It's billions of asynchronous individual fates all just averaging out into one single line.
And validating that analogy, treating the phases as a statistical distribution of single-cell behaviors rather than a synchronized group activity that completely changes how we approach medicine.
How so?
Well, when you understand that a population is asynchronous, you suddenly grasp the mechanism behind why an antibiotic almost never kills 100% of an infection instantly.
Oh, well, because they aren't all doing the same thing.
Right, because at any given second, even in the middle of a massive explosive growth phase, a small subset of individual cells might just be sitting there, not actively dividing. They are the statistical outliers in your traffic jam analogy.
That is fascinating. And, that statistical nature naturally leads to a historical question. Like, who actually did the math to establish this model in the first place?
Yeah, that's a great question.
Looking through the research papers we have, the modern interpretation really traces back heavily to this French biologist, Jacques Monod. He basically made bacterial growth curves the entire subject of his 1941 doctoral thesis. And then he published this landmark paper in 1949.
Yeah, Monod is an absolute giant in this space. I mean, obviously he didn't discover that bacteria multiply, we knew that. But his genius was taking a purely descriptive curve and engineering it into a predictive mathematical model.
Which is a huge leap.
A massive leap. And he accomplished this through what we now call the Monod equation. He figured out how to directly link the exponential growth rate of a population to the concentration of a single limiting nutrient in the environment.
So by doing that, he essentially proved that as the food source depletes, the growth rate doesn't just, randomly slow down. It drops predictably according to a very specific calculable constant.
Exactly. He basically gave birth to bacterial growth kinetics.
But, and I found this super interesting in the sources, if you look back even earlier, there's an American bacteriologist named Robert Earl Buchanan who was working on this back in 1918.
Oh, yeah, Buchanan.
Yeah, and Buchanan heavily criticized the impressionistic descriptions of his peers. He didn't want the 4 broad phases we use today. He actually proposed a really rigorous 7-phase model.
Right, he had all these transitional phases.
Exactly. He included an acceleration phase, a deceleration phase, all these highly specific transitional steps. So wait, if Buchanan had a mathematically sound 7-phase model, why do we only teach 4 today? Didn't we lose some critical nuance by simplifying it?
Well, what's fascinating here is that the reduction to 4 phases was driven almost entirely by pedagogical and practical limitations.
So it was just easier.
Basically, yeah. 4 phases are just simply easier to teach, they're easier to memorize, and fundamentally they're way easier to test on an exam.
That makes total sense.
Plus, in a laboratory setting, physically measuring those tiny, fleeting transitional phases, it requires taking samples almost constantly, which is incredibly labor-intensive.
Right.
But biologically speaking, Buchanan was absolutely right. The transitions are vital because biology does not operate like a light switch. A bacterial population's growth rate doesn't just snap from absolute zero to maximum overdrive instantly.
Right, because spooling up the actual biological machinery takes time. Which really brings us to the actual journey of the bacteria. Let's trace their path through a closed system, starting from the exact moment they are dropped into a fresh, nutrient-rich broth.
The starting line.
Exactly.
Yeah.
This is phase 1, the lag phase, the warm-up period. And the most critical correction the sources make here is shattering the idea that lag means dormant.
Yes, that is such a common misconception.
When bacteria hit a new food source, the cell number doesn't rise immediately, so the graph is totally flat. It looks like they're doing absolutely nothing, but internally,
Yeah.
they are metabolically hyperactive.
Oh, they are frantically remodeling their internal architecture just to survive and exploit this new environment. When a cell enters a new medium, it has to uncoil its DNA, synthesize vast amounts of messenger RNA, build new ribosomes.
Just a massive construction project.
Exactly. It has to repair any environmental damage from the transfer and manufacture highly specific enzymes tailored to whatever new nutrients they've just encountered. And all of that molecular manufacturing requires massive amounts of energy, which is exactly why they aren't dividing yet. They are building the factories required for division.
And the sources provide this brilliant, tangible example of this with Listeria monocytogenes, which actually explains the exact mechanism behind how your refrigerator works and why your milk spoils.
Oh, the fridge example is perfect for this.
Right. So if you take a population of Listeria from a cold, 4°C environment, like the inside of your fridge, and you introduce it to a room temperature environment, it will lag for many hours, sometimes over a full day.
Because it's completely unprepared for the heat.
Exactly. It
Yeah.
literally has to manufacture entire suites of new temperature-appropriate enzymes before it can safely initiate cell division.
And the entire field of food safety and the calculation of use-by dates rely heavily on this specific metabolic delay.
Really?
Yeah, the primary goal of standard cold storage isn't to actually sterilize the food, nor does it completely halt bacterial metabolism forever.
It just slows them down.
Exactly. The engineered goal is to artificially stretch that lag phase out for as long as functionally possible.
But eventually that remodeling finishes. The new enzymes are built, the factories are online, and the brakes come off. The population aggressively enters phase 2, which is the log or exponential growth phase.
This is where things get crazy.
The cell number begins to double at a constant interval known as the generation time.
And the sheer speed of biology during this phase is staggering. Under optimal laboratory conditions at 37°C Escherichia coli, or E. coli, has a generation time of roughly 20 minutes.
20 minutes.
Yeah. It is among the fastest documented doubling times for any free-living organism.
And here's where it gets really interesting. The mathematical implications of that 20-minute cycle are borderline incomprehensible.
They really are.
The sources include this classic microbiology shock fact to illustrate this. If you take just one single E. coli cell and you allow it to divide unrestricted every 20 minutes for just 24 hours, you get 72 doubling events, 2 to the power of 72.
Which is a very big number.
Yeah, it results in roughly 4.7 times 10 to the 21st power individual cells. If you calculate the physical mass of those cells, that single day of growth would theoretically weigh vastly more than the entire planet Earth.
It is a phenomenal demonstration of exponential power. But, obviously we aren't currently standing on a giant planetary ball of E. coli.
Right. So the obvious question for you is, how does nature prevent that? How do they self-limit before completely consuming the globe?
Well, unbounded exponential growth is basically a mathematical fantasy because the very act of exploding in population size aggressively alters their local environment.
They ruin their own home.
Exactly. They consume the available nutrients at this terrifying, accelerating rate, and simultaneously they excrete massive amounts of metabolic waste like complex acids or alcohols, which rapidly alter the pH of the fluid and toxify their own environment.
Oh, so they are poisoning themselves while starving.
Exactly. Combine starvation and toxicity with literal physical crowding, and the environment violently forces the population out of the log phase.
But before we move out of that explosive phase, there is this monumental historical experiment we definitely need to cover. The sources detail Salvador Luria and Max Delbrück's famous fluctuation test from 1943. Which actually eventually won them a Nobel Prize.
Oh, the Luria-Delbrück experiment. Classic.
They proved that random resistant mutant bacteria arise naturally during this normal log phase growth, long before the bacteria ever even encounter a threat.
Yeah.
And the mechanism of how they proved this is just genius.
It really is elegant.
They grew multiple, separate parallel cultures of bacteria, and at the very end of the growth phase, they exposed all the tubes to a deadly virus, a bacteriophage. If the bacteria only mutated in response to the virus, every tube would have a roughly similar small number of survivors.
Because the exposure would be the trigger for the adaptation.
Sorry, I mean, that would be the logical assumption.
Right.
But that's not what happened. Luria and Delbrück found massive fluctuations. Most tubes had 0 survivors, but a few tubes had absolute jackpots, like thousands of resistant bacteria.
And that mathematical variance proved that the mutation for virus resistance happened entirely by random chance early in the log phase, generations before the virus was ever introduced.
It's incredible.
It firmly established the bedrock of modern evolutionary biology. Resistance is not some proactive choice a bacterium makes when it gets attacked. Mutations are simply replication errors that occur naturally during that rapid 20-minute doubling cycle.
So they're just lucky mistakes.
Exactly. The genetic variations are already present in the population, silently riding along, just waiting for a shift in the environment to suddenly become advantageous.
And that environmental shift happens abruptly when the population hits the wall we mentioned earlier. Phase 3, the stationary phase.
The wall.
The nutrients deplete, the toxic waste builds up to critical levels, and rapid division just screeches to a halt. The growth rate falls to 0. So the number of cells successfully dividing perfectly matches the number of cells dying, which causes the total population to plateau. If you look at the graph, this actually resembles the logistic growth model that's commonly used in macroecology.
And just like the lag phase, the stationary phase is widely misunderstood because a flat line implies biological silence.
Right, it looks like nothing is happening.
But in reality, the cells are in a state of absolute physiological panic. They are radically reprogramming their internal systems for long-term survival. In E. coli, this panic response is largely governed by a specific gene that produces an alternative sigma factor called RPOS.
And the mechanism of how that works is incredible. A sigma factor essentially acts like a steering wheel for the cell's DNA reading machinery, the RNA polymerase.
Good analogy, yeah.
RPOS physically grabs the machinery, drives it away from all the genes responsible for growth and division, and forces it to transcribe stress survival genes. It forces the cell to build thicker peptidoglycan walls, cross-link its membrane proteins, and basically batten down the hatches against the incoming famine.
It is a total shift from a growth-oriented metabolism to a maintenance-oriented metabolism. And this environment is vicious. The sources highlight Roberto Coulter's discovery in the 1990s, a phenomenon known as JAS-SP.
which stands for growth advantage in stationary phase.
Exactly.
JAS-P operates like this microscopic, hyperlethal game of musical chairs. The total number of chairs, meaning the remaining resources in the closed system, is completely locked.
Right. No new food is coming in.
The total population size isn't growing. But those random mutant subpopulations that arose back in the log phase, they are now aggressively scavenging. The mutations allow them to catabolize the remains of their dead neighbors more efficiently.
It gets really dark.
It does. They continuously cycle through waves of selection, vigorously outcompeting and cannibalizing the original population. The overall cell count on the graph barely moves, but underneath that flat line, it is literally an evolutionary bloodbath.
And human industry has actually learned to exploit this specific panic response.
Really? How so?
Well, when pharmaceutical companies manufacture antibiotics, or even when breweries ferment certain beers, or artisans craft complex cheeses, they deliberately time their biological harvest to this stress-induced stationary phase.
Wait, they wait for the panic.
Exactly. During the log phase, bacteria are only focused on primary metabolism, making more cells. But when they are starving and stressed in the stationary phase, they begin producing secondary metabolites.
Ah, I see.
These are complex chemical weapons and defenses they secrete to secure those limited resources. We literally harness their chemical warfare for our own use.
That is wild. But eventually, the toxicity overwhelms even the best defenses, pulling the population into phase 4, the death phase, or decline. The viable cell counts finally begin to fall, usually at a logarithmic rate, although the sources note it's a much slower curve than the explosive upward trajectory of the log phase.
Yeah, they succumb to severe starvation. But increasingly, molecular evidence shows this isn't just passive withering. They actually actively dismantle themselves through antitoxin systems.
And the mechanism there sounds straight out of science fiction. The bacterial chromosome encodes a highly stable toxin and a very unstable antitoxin.
Right.
During healthy growth, the cell constantly produces both, and the antitoxin neutralizes the toxin.
It keeps it in check.
Right, but under the severe stress of the death phase, the cell stops producing the antitoxin. Because the antitoxin is unstable, it rapidly degrades, leaving the stable toxin free to destroy the cell from the inside out.
It's a literal active self-destruction sequence.
The curve spirals down towards zero. Game over. But, and this is a huge but, this is where the historical texts throw a massive curveball into the conventional model.
Right, because the textbook model demands an inevitable end, but biological reality presents the undead exceptions.
The undead exceptions. There are two monumental debates detailed in the sources that fundamentally changed how the medical community views infectious disease. The first is the VBNC state. Viable, but non-culturable.
This one is fascinating.
Back in the 1980s, microbiologist Rita Colwell was studying Vibrio cholerae, the pathogen responsible for cholera, resting in cold marine water. Standard Petri dish assays showed a textbook death phase. The bacteria were just disappearing. But Colwell discovered these supposedly dead cells were actually still viable.
And the mechanism behind this non-culturability is just fascinating. Standard laboratory Petri dishes are packed with rich, heavy nutrients.
Right, it's an all-you-can-eat buffet.
Exactly. But when you take a starved, highly stressed Vibrio cell and suddenly drop it onto that buffet of rich media, the sudden, overwhelming influx of nutrients causes fatal oxidative stress.
Oh, wow, so the food kills them.
The laboratory test itself was killing them. They couldn't form colonies on a plate. But if those exact same dormant cells were ingested into the gradual protective environment of a human host, they could completely resuscitate and trigger a cholera outbreak.
Which, naturally, sparked intense debate. Critics in the late 90s, like Barer and Harwood, argue that many of these VBNC cells were truly just in a state of slow, irreversible decay, not some specialized dormancy.
There was a lot of pushback.
But regardless of where scientists land on that spectrum, the debate forced this deeply uncomfortable realization. Just because nothing grows on a lab plate does not mean the pathogen is actually eradicated.
Unculturable is definitely not synonymous with dead.
Which flows directly into the second and perhaps more universally relevant exception, persister cells.
Oh, yes.
Joseph Bigger documented this phenomenon way back in 1944. He treated a culture of Staphylococcus with penicillin. Every single cell in this specific culture was genetically identical, and they were all entirely susceptible to the drug. Yet, a microscopic fraction of the population somehow survived the chemical onslaught. He named them persisters.
And for decades, persisters were just dismissed as a bizarre laboratory artifact, just a glitch. Today, they are recognized as the primary mechanism behind chronic, recurring illnesses, from tuberculosis to stubborn cystic fibrosis lung infections to biofilms on medical implants.
Now, I want to clarify a vital point here for anyone listening who might be a bit confused by the terminology and the sources. Are these persister cells the exact same thing as the antibiotic-resistant mutant bacteria we talked about earlier?
If we connect this to the bigger picture, the answer is no, absolutely not. And the distinction is paramount to modern medicine.
Okay, lay it out for us.
Persister cells are absolutely not resistant mutants. If you isolate a surviving persister cell, place it in fresh broth, let it multiply, and then hit its offspring with that exact same antibiotic, the entire offspring population will die.
Oh, so they don't pass on immunity.
No, they hold no genetic immunity whatsoever. Persisters are phenotypic variants.
Meaning they survive because of what they aren't doing. Penicillin and a lot of major antibiotics operate by targeting active cellular machinery, specifically the enzymes that build the bacterial cell wall.
Right.
So if a bacterial cell stalls its own metabolism and enters a dormant, non-growing state, it just stops building its wall. The antibiotic rushes in, but there is no active machinery to jam up. The drug simply floats by harmlessly.
Exactly. An antibiotic compound can perform with 100% efficacy in a controlled laboratory environment against a specific bacterial species and still completely fail to cure the infection in a human patient.
Because the drug fundamentally requires the bacteria to be actively growing in order to kill them.
Exactly.
And this single mechanistic reality explains the doctor's lecture from the very beginning of our deep dive.
Yes, the militant lecture.
When you take the first 3 days of an antibiotic prescription, you effectively slaughter the massive, actively dividing log phase population. The sheer bacterial load drops, your immune system finally catches its breath, and you feel physically better.
You feel cured.
But hidden deep in the wreckage are those dormant persister cells. If you start taking the medication early, the antibiotic clears from your bloodstream. The persisters eventually wake up, recognize the environment is safe, flip their machinery back on, re-enter the log phase, and you suffer a massive relapse.
You have to maintain the chemical pressure long enough to catch the persisters as they eventually attempt to wake up and divide.
It's all about outlasting them. So pulling this all together, the bacterial growth curve is far more than just a static picture of a population. It's an active, pulsating distribution of cells.
It really is.
They rush from the hyperactive internal remodeling of the lag phase into a world-consuming exponential explosion in the log phase. They hit the stationary wall, triggering the brutal molecular scavenging of GSP, and finally, they execute specialized self-destruction or hide out as undead persisters during the death phase.
It is the complete life cycle of an entire microscopic civilization.
And the invisible rules governing that civilization dictate how we preserve our global food supply, how we synthesize our most vital medicines, and how we clinically fight infectious disease.
But before we wrap up, the sources do leave us with one final kind of mind-bending concept.
Oh, yes.
We've spent this entire time analyzing a closed system, a batch culture with a finite beginning and an inevitable end. But human engineering actually found a way to cheat the system entirely.
Yes, the invention of the chemostat.
The chemostat, exactly.
Invented around 1950 by Jacques Monod, alongside Aaron Novick and Leo Szilard, the chemostat is effectively an artificial mechanical stomach.
That's a great way to put it.
It is designed to continuously drip fresh liquid nutrient medium into a culture vessel at a highly precise rate, while simultaneously draining away the exact equal volume of toxic waste in older cells.
It is a masterclass in manipulating biological kinetics. They looked at the natural mathematical law of the growth curve, the inevitable decline caused by starvation and toxicity, and they built a machine to functionally erase it.
They broke the rules.
By perfectly balancing the dilution rate of the fresh food coming in with the specific growth rate of the bacteria, a chemostat suspends the bacterial population in an infinite, theoretically immortal state of log phase exponential growth.
So the four-phase curve is a fundamental rule of nature. But human ingenuity managed to engineer a loophole. That is an incredible thought to ponder. Thank you for joining us on this deep dive into the microscopic world. We encourage you to think just a little differently the next time you glance at the expiration date on your milk, or, the next time you reach into your medicine cabinet.
The invisible war zone is always raging.
Catch you next time.